Hello, my name is Dakota Archer, and I'm going to give a brief overview on cellular respiration for Anatomy and Physiology 1. Cellular respiration is essentially the process that utilizes specific molecules and enzymes to manufacture adenosine triphosphate, or ATP. The first step in this process is known as glycolysis, and it occurs in the cytosol. The process begins with a 6-carbon molecule of glucose, and with 2 ATP molecules from the previous cellular respiration cycle pairing with digestive enzymes, begins to break down this 6-carbon molecule of glucose and split it into 2 molecules of pyruvate. So during this stage, energy is produced, which is then used to turn 2 adenosine diphosphate molecules, ADP, into 2 ATP molecules. This is done by adding a phosphate group to the ADP molecules. And it's worth noting that NAD+ is also converted to NADH during this stage, which will later be used to create more ATP molecules. ATP molecules are essentially our cells' energy sources to carry out their essential functions. Now the NADH that's created will also carry the electrons up and out of this area, and then we'll move on to the Krebs cycle. So after glycolysis, the next step is known as the Krebs cycle, like I said. It's also known as the citric acid cycle. So during this stage, the 2 pyruvate molecules that were created during glycolysis are individually converted to acetyl coenzyme A, or acetyl-CoA. The process occurs in the mitochondrial matrix. The matrix is the fluid component that surrounds the mitochondrial structures. So the 2 pyruvates are broken down into acetyl-CoA and carbon dioxide gas. Energy from this breakdown is also used to convert 2 NAD+ molecules into NADH. And then the acetyl-CoA is then continues to break down further until basically only carbon dioxide remains. So energy from the bonds is used to convert NAD+ and FADH+ into NADH and FADH2. 2 ATP molecules are also produced from the 2 ADP molecules during this stage. Electrons are then carried out by NADH and FADH2. And we move on from the Krebs cycle to the electron transport. So this is the final stage of cellular respiration. This occurs within the mitochondrial membrane. So this is the mitochondrial membrane, and this is the matrix. All right, so what we have is protein complexes that are embedded within the membrane. And this is the stage where all the molecules from the glycolysis and the Krebs cycle are finally put to use. So NADH and FADH2 produce high energy electrons that will allow these protons to be pumped out of the matrix through the membrane. And this happens because the energy, the electrons created embed themselves within this membrane, and when they move across it, they create a chain reaction, forcing the protons out. Now, the protons will be going against the concentration gradient, going from a low concentration to a high concentration. And once all the protons are pulled from the matrix, the large accumulation will lead to rapid diffusion through the ATP synthase. The hydrogen protons activate ATP synthase. So as the protons diffuse back through into the matrix through ATP synthase, ADP will bond with phosphate, creating ATP. And this process will occur many times until upwards of 34 to 36 ATP molecules are created from one 6-carbon molecule of glucose. Now, to keep the process efficient and remove any hydrogen and electrons that have accumulated, oxygen will enter and bond with 2 hydrogens and 2 electrons, creating H2O, water. And then the process will continue. ATP molecules from this stage will carry over to the beginning of a new cellular respiration cycle, breaking down glucose molecules and resuming the process over. All right, thank you very much.